Hi, this is Dr. Ikuo Tsunoda, Louisiana State University. Before I relocated my laboratory to Louisiana, I have investigated multiple sclerosis in Salt Lake City, Utah. Back then, when I had guests, I usually showed them around Temple Square. This is one of such occasions. In Utah, Mormon Church is usually called LDS, which stands for the Church of Jesus Christ of Latter-day Saints. In this section, let's discuss about the role of axonal degeneration in an autoimmune model for multiple sclerosis, EAE. To investigate the role of axonal degeneration, we used a mutant mouse strain called C57 black wlds mice. wlds mice are a substrain of C57 black 6 mice. wlds stands for Western Latter-day Saints? No, it is Wallerian degeneration slow. I will refer to as wlds mice. wlds mice are protected from Wallerian degeneration by overexpression of a fusion protein encoded in a chimeric gene WLD. WLD encodes complete sequence of nicotinamide mononucleotide adenine transferase and N-terminal 70 amino acids of ubiquitin fusion degradation protein 2, UFD2. Since NMNAT is a synthetic enzyme of NAD, this will increase NAD concentration, or this further increases a downstream enzymatic activity of SIRT1. The mechanism of axonal protection is still controversial. It can be due to an increase of NAD or SIRT1 or UFD2 or a combination of all. Right now, please just remember this NAD synthetic enzyme NMNAT and SIRT1. In wlds mice, transected axons can survive or function for weeks. What happens if axons are cut? When axonal transection occurs in wild-type mice, for example, a parent strain of wlds mice B6 mice, the distal stump of axons will degenerate, and we will see axonal swelling or loss. This is Wallerian degeneration. This degenerated axon is detected by immunohistochemistry against non-phosphorylated neurofilament. On the other hand, when the axon is cut in wlds mice, this transected axon can survive and function for weeks and is negative for non-phosphorylated neurofilament staining. Using wlds mice, we induced EAE by subcutaneous injection of myelin oligodendrocyte glycoprotein MOG 35 to 55 peptide. In EAE, MOG-specific autoimmune T cells initially attack myelin sheaths. Severe inflammation damages axons secondarily, with eventual distal axonal degeneration, and this would lead to disease progression in B6 mice. But what happened to wlds mice, which will not develop axonal degeneration? We induced EAE using MOG 35 to 55 peptide in wlds mice and its parent strain B6 mice. On your left, you can see clinical scores of EAE. During the first 2 weeks, the onset and progression of EAE were similar between the parent strain B6 shown in black, and wlds mice, shown in red. However, from this point, wlds mice did not show disease progression and recovered completely. On the other hand, B6 mice developed severe paralysis at 3 weeks and remained paralyzed during the entire 2-month observation period. On your right is anti-MOG autoimmune T-cell responses against MOG. Similar to clinical signs, at 2 weeks, both mouse strains showed the same levels of anti-MOG T-cell responses. However, the responses in wlds mice, shown in red, declined thereafter. On the other hand, B6 mice showed enhanced anti-MOG T-cell responses. So clinical signs and autoimmune T-cell responses associated well. This is the ventral funiculus of the spinal cord stained with luxofast blue. On your left, B6 mice, and on your right, wlds mice. At disease onset at 2 weeks, both mouse strains developed similar inflammatory lesions. You can see similar mild meningitis here in both mouse strains. However, at 3 weeks, extensive demyelination is seen as white vacuolar lesions only in B6 mice. Here, inflammation is also more severe than that of 2 weeks. On the other hand, wlds mice showed a similar pathology to that of 2 weeks, and demyelination is mild, if any. How about axons? Axonal degeneration was detected by immunohistochemistry against non-phosphorylated neurofilament. Damaged axons are seen in brown dots. The top 2 panels are axonal damage in spinal cord of B6 mice. We can see high levels of axonal swelling positive for neurofilament at 2 and 3 weeks. In contrast, in wlds mice, axonal degeneration was slow. We do not see axonal degeneration at 2 and 3 weeks. 1 month after, axonal degeneration is detectable. Only after 2 months, we can see extensive axonal degeneration. So, axonal degeneration is really slow in wlds mice. This is what most likely occurred in the EAE model. In wild-type mice, after induction of EAE, mice develop anti-myelin immune responses that attack myelin sheaths from the outside, and severe inflammation damages axons and causes Wallerian degeneration of distal axons. Then, myelin sheaths further degenerate and myelin antigen is presented to T cells and enhance anti-myelin specific autoimmune responses. This results in disease progression. However, in wlds mice, since Wallerian degeneration did not follow here, it prevents a cascade reaction. As a result, we did not see enhancement of anti-myelin responses or disease progression. Does this mean that keeping axons is always good? So we decided to look at another animal model, Tyler's virus infection.